Every single day, somewhere in the world, a sequence of events begins that no one notices until it is already too late. It does not start with violence. It does not start with chaos. It starts with something ordinary, a routine task, a familiar environment, a decision made in a single second that seems completely harmless. And then, without warning, the human body begins to fail in ways that science has spent decades trying to understand. This is the story of how one mistake, repeated across history in laboratories, operating rooms, industrial sites, and quiet homes, has revealed one of the most terrifying truths about the human body, that it is far more fragile than we believe, and that death, when it comes from a single error, does not always announce itself with pain or panic. Sometimes it is silent, sometimes it is invisible, and sometimes, by the time anyone realizes what has happened, there is nothing left to do but watch and record what the body does when its most fundamental systems collapse one by one. To understand how one mistake can end a life, you first have to understand how the human body keeps itself alive. Right now, as you watch this, your heart is beating somewhere between 60 and 100 times per minute. Your lungs are pulling oxygen from the air and pushing it into your blood. Your liver is filtering toxins. Your kidneys are processing waste. Your brain is monitoring all of it simultaneously, firing electrical signals across billions of neurons, adjusting and correcting and compensating every fraction of a second. The human body is not just a biological machine, it is the most sophisticated system ever assembled in the known universe, and it works because of an intricate web of chemical and electrical processes that depend on each other in ways so complex that even now, in the 21st century, scientists and doctors are still discovering new layers to its design. But here is the terrifying truth that sits at the heart of everything you are about to hear. That same complexity, that same beautiful intricacy, is also the reason one mistake can bring it all down. Because when you have a system where everything depends on everything else, a single point of failure does not stay small, it spreads, and it spreads fast. The year is 1994. Karen Wetterhahn is a chemistry professor at Dartmouth College in New Hampshire, and by every measure she is one of the most careful, most disciplined scientists in her field. She has spent years working with toxic compounds, following strict safety protocols, wearing protective gear, operating inside ventilated hoods. She is not reckless. She is not careless. She is, by every account of her colleagues, exactly the kind of scientist who takes danger seriously. On a quiet afternoon in August, she is working in her laboratory with a compound called dimethylmercury, a form of mercury used in chemical research as a calibration standard. She is wearing latex gloves, she is working carefully, and then a single drop of the liquid falls. It lands on her gloved hand. She wipes it away. She continues working. What Karen Wetterhahn does not know in that moment, what almost no one in the world knows at that point, is that dimethylmercury does not behave like other toxic substances. It does not wait at the surface of a material. It does not get stopped by a glove. It passes through latex in less than 15 seconds, and it is absorbed directly into the skin. In the space of a single heartbeat, an invisible poison is already moving through her body, and she feels nothing at all. The weeks that follow are unremarkable. Karen goes home. She teaches her classes. She attends meetings. She works in her lab. She laughs with her colleagues. She does everything she normally does, and she feels fine, because that is what makes this particular poison so uniquely devastating: the delay. Dimethylmercury crosses the blood-brain barrier, the body's own protective wall between the bloodstream and the brain, and it begins to accumulate in the central nervous system. But it does this quietly, invisibly, while the rest of the body compensates and the mind remains unaware. The scientific community has a name for this kind of period. They call it a latent phase. The body has been compromised, but the damage is building in a place where the early warning signals are too subtle to recognize, hidden beneath the noise of ordinary daily life. And so Karen continues living her life, completely unaware that inside her skull, mercury is slowly destroying the neurons she has spent a lifetime building her career around. It is not until 5 months later, in January of 1997, that the first signs appear. Karen begins to feel slightly unsteady. She notices small problems with her balance. She has trouble articulating certain words. At first, these symptoms seem minor, the kind of thing you might dismiss as tiredness or stress, but over the following weeks and months, they accelerate. Her vision begins to narrow. Her speech becomes harder to control. Her movement becomes uncoordinated. And then her doctors run blood tests, and the results come back with a mercury level in her blood that is more than 80 times the toxic threshold. 80 times. The number is so staggering that some of the physicians initially assume there has been a laboratory error. There has not. By the time Karen Wetterhahn is admitted to hospital, the damage to her neurological system is already catastrophic and irreversible. Dimethylmercury had not just poisoned her brain, it had dismantled it, systematically destroying the regions responsible for coordination, speech, vision, and memory. She slips into a coma. On June 8, 1997, less than a year after a single drop of liquid landed on her glove, Karen Wetterhahn dies. She is 48 years old. Her death changes the safety protocols for an entire field of science, and it teaches the world something devastating about the gap between what we believe protects us and what actually does. But the story of how one mistake turns fatal is not only about poison, it's about the body itself, about the precise choreography of organ systems that most of us have never had to think about because they have always worked perfectly. Consider the heart. The human heart is a muscle, and like all muscles, it runs on electricity. A cluster of cells in the upper right chamber, called the sinoatrial node, generates a tiny electrical impulse roughly once every second, and that impulse travels down through the heart in a specific, exact sequence, causing the chambers to contract in perfect coordination. First the upper chambers, then the lower, over and over, 60 to 100 times per minute, every minute of every day for the entirety of a human life. The system is extraordinarily reliable, but it is also extraordinarily sensitive. The electrical signals that govern the heartbeat depend on the movement of specific ions, sodium, potassium, calcium, in and out of heart cells through tiny channels in the cell membrane. And when something disrupts that movement, even slightly, the consequences cascade outward with breathtaking speed. This is precisely what happened in a case that became one of the most studied events in sports medicine history. In 1998, a young basketball player, healthy, athletic, with no known heart condition, collapsed during a game after taking a blow to the chest from a ball. He had not been hit hard. The ball had struck him at the exact moment between heartbeats, in a window so narrow that scientists who later analyzed the physics of the event determined it lasted less than 15 milliseconds. But that tiny window was enough. The impact disrupted the electrical signal at the one moment when the heart's cells were in the most vulnerable phase of their cycle, triggering a catastrophic arrhythmia called ventricular fibrillation, a state where instead of beating in coordinated rhythm, the heart simply quivers. Hundreds of individual muscle fibers contracting randomly, independently, without coordination, generating no actual pumping force at all. In medical terms, this is cardiac arrest. In practical terms, it means the brain stops receiving oxygen in seconds, and without intervention, irreversible brain damage begins in 4 minutes. The young man survived only because a defibrillator happened to be nearby and was used within the critical window. His case gave a name to the phenomenon, Commotio cordis, from the Latin for disturbance of the heart. It now accounts for roughly 20% of sudden cardiac deaths in young athletes. One ball, one blow, one 15-millisecond window, one mistake in timing made not by a human being, but by physics, and the most powerful muscle in the human body simply stops working. Understanding the mechanics of these moments, the way one wrong element in one wrong time produces total systemic collapse, requires stepping back and looking at what scientists call the cascade of failure. The human body does not simply stop all at once. It fails in sequence, with each failing system triggering the next, like dominoes falling in slow motion. Take the example of blood pressure. When the heart stops pumping effectively, blood pressure drops. When blood pressure drops, the body responds by constricting blood vessels in the extremities to try to maintain pressure in the core, the brain, the heart, the lungs. This is an emergency survival mechanism, an ancient biological response that has kept human beings alive through millions of years of evolution. But if the underlying cause is not corrected, the constriction only buys time. Blood flow to the extremities is reduced, cells begin to suffer from oxygen deprivation, and the kidneys, which depend on blood pressure to filter waste, begin to fail. As